Hello everyone, my name is Chen Ke Hao. Today I will present on the topic application of nanotechnology in plant genetic engineering. This article, this article was published in the International Journal of Molecular Science on October 2023 with an impact factor of 4.9. The content is closely related to the nucleus and genetic materials. The relevance of this article to my topic lies in the fact that nanotechnology provides a means to overcome the barriers of the cell wall and the nuclear membrane. In a broadly exogenous DNA, RNA, and proteins to infiltrate into the nucleus and directly act on genetic materials for gene editing or impression regulation. With the continuous growth of the global population, food demand is steadily increasing, compelling agriculture to adopt more advanced technologies to enhance core yields, strengthen resilience against extreme and unpredictable supply, and reduce loss caused by pests and disease. Genetic engineering technologies have been widely applied to various plant species, with the CRISPR engineering as the latest mainstream approach. A core challenge in genetic engineering lies in the efficient delivery of gene cascades into plant cells. Traditional gene delivery methods are often time-consuming, labor-intensive, prone to tissue damage, and lived by low transformation efficiency. And in contrast, nanoparticle mediated gene delivery demonstrates higher efficiency and broader adaptability across the species. This review summarizes the evaluation of biomolecular delivery methods in plants, focusing particularly on nanotechnology, including nanoparticle types, preparation strategies, transport machines, and their advanced application in plant genetic engineering. Introduction: Global population growth, climate change, and COVID-19 have intensified pursuit on agriculture and food security, making biotechnology crucial to improve crop yields and resilience. Gene editing, especially CRISPR-Cas9, plays a key role in enhancing stress tolerance, yet many plants remain difficult to transform due to cell wall barriers and low inflation. Conventional methods such as a growth bacterial gene gun, adaptive population, and PG phase leaving tension like low efficiency and tissue damage. In contrast, nanotechnology offers tunable nanoparticles capable of penetrating cell walls and membranes to infiltrate dividing DNA, RNA, or proteins. This review highlights nanotechnology's advantages in gene material divider, plant rejuvenation, and gene editing, while also addressing safety concerns. Traditional plant genetic transformation methods. Traditional plant genetic transformation has advanced biology, but face limits from cell walls and low efficiency. Genes can divide DNA code particles across barriers, but cause tissue damage and requires costly equipment. A broad battery integrates tDNA infosynthesis and stability, yet is restricted to DNA delivered host range and raise GM concern. Electroporation use activated fields for high efficiency molecular entry, but fixed cell walls reduce effectiveness and may damage cells. PG devices is simple, efficient, and low cost, for leads to photopress and mainly for transient impression. Nanoparticle mediate biomolecular devices. Nanoparticles are artificially manufactured particles smaller than 100 nanometer with temporal, physical, and chemical properties that enable precise interaction with biomolecules. They have energy as custom sample, high potential tools widely applied in medicine, and agriculture, attracting strong scientific interest in manual systems. Engineered nanoparticles can carry molecular cargo or relieve proteins at the subcellular level, avoiding toxins dividing to the nucleus or organelles. This knowledge also informs plant transformation, supports cross-breeding and resilience. Importantly, studies show that nanoparticles can enter cells without external force, overcoming key limitations of traditional transformation methods. Types of nanoparticles, including DNA nanostructures, particles, silica, clear polymers, and metals, possess distinct properties, enabling transient or stable transformation. In plant delivered magnetic nanoparticles can introduce DNA into pollen to form transgenesis. For infections, is debate. Nanobases have delivered genes into messy pollen, and carbon nanotubes can penetrate cell walls to transport plasmid DNA or small RNA as well. Prevention degradation. Most nanoparticles mediate deliver leads to transient expression without genuine integration, offering simplicity, short cycles, high efficiency, and biosafety, while potentially bypassing GMO regulations and easing food safety concerns. Methods for nanomaterial preparation. Nanomaterials can be prepared by physical, chemical, or biological methods using two main strategies. top-down, which breaks both bulk materials into small particles, and bottom-up, which builds from small nanostructures usually chemically. Green synthesis, with plants exchange or microbes is engineered using ions like Ag, Cu, Co, and Ni from metal oxide nanoparticles. This approach lowers costs, reduces toxicity, and is more eco-friendly. Particle size, shape, and properties affect transport efficiency, and normal distances matters that are efficiently simple and pollution-free may provide ideal platforms for intracellular biomolecular delivery. Manners for nanomaterials preparation. These nanoparticles have diverse properties and can be prepared and delivered from plant transformation. They enter cells via diffusing, endocytosis, plasma desmata, or by causing virus with physical and chemical manners, delivering DNA, RNA, or proteins from transient or stable transformation. Targets include mature plants, which absorb nanoparticles mainly for leaves and loops before vascular transport, and the suspensing cells. Compared with traditional methods, nanoparticle mediated transformation offers clear advantages. The absorption and transport of nanoparticles in plants are influenced by factors such as size, shape, and charge. The many interfloral leaves and roots and can overcome barriers by diffusing endocytosis or plasmatismata. Traveling long distance for slim plastic and upper plastic pathways. Although their movement within plants has been confirmed, the extreme machines remain unclear, requiring further research to advance genetics, engineering, and agricultural nanotechnology application. Application nanotechnology in plant genetic transformation. Nanotechnology enhances plant genetic transformation by using nanoparticles as small, biocompatible, and temporal carriers. They protect DNA, siRNA, and proteins, enabling gene transfer, silences, and DNA-free editing. Carbon nanotubes and DNA nanostructures have delivered genetic cargo into nuclear and chloroplasts. Targets include mature plants, suspension cells, and protoplasts. For original entry machines and protoplasty regeneration remain major challenges. Nanotechnology improves plant gene editing by using nanoparticles as small biocompatible carriers that protect DNA, siRNA, and protein. They enable precise CRISPR-Cas9 delivery, gene transfer sequencing, and DNA-free editing. Carbon nanotubes, lipid nanoparticles, and DNA nanostructures have been used to deliver genetic cargo into nuclei and chloroplasts. Despite challenges like orogeny and and plastic regulation, this approach shows strong potential to advance crop breeding. Fusion integrating gene editing, nanotechnology, and de novo regeneration could transform plant engineering. More research is needed to design nanomaterials for efficiency, delivery, and breeding. Current methods rely on gene guns or electromagnetic fields with low efficiency, while CRISPR delivers fast of target risk. Nanoparticle architect and transport remain unclear. And most transformations are transient, require improved regulation for stable crops. The extract the illustrates a plant transformation method that integrates gene editing, nanotechnology, and de novo regulation. Multiple gRNAs are first assembled into CRISPR methods and then delivered into intact plant meristem, ultimately producing transgenic plants without reliance on tissue culture. Perspectives cause disciplinary research is needed to study nanomaterials plant interaction. AI and deep learning can improve gene prediction. Closed tissue may add nanoparticle deliver. Safety and off-target risks must be assessed with long-term monitoring of GNOS and nanomaterials. Future work should develop safer nanocarriers to support breeding and food production. This is my result. Thank you for your listening.